The first thing to know about viruses is that they're not technically alive. So if you remember back to those criteria of life that we talked about at the very beginning of this semester, viruses are not going to meet some of those criteria. Just to give you a couple of examples, they're not made of cells. They're not composed of cells. They're not organized at the cellular level. They can't carry out the chemical reactions of metabolism on their own or reproduce without a host cell. So because they are not alive or non-living, we refer to them as particles. So it's a non-living particle that at its most basic consists of a genome, so genetic material, enclosed by a protein coat. So your most basic virus has some sort of protein coat called a capsid, and inside of it, it has genetic material. Some viruses will be more complicated than this, but that's kind of the basic material. There's over 4,000 different types of viruses that have been identified, and they're going to vary a lot. So again, we have sort of this basic structure, but they're going to have a lot of differences in what that capsid looks like, what the genetic material is made of, and what kind of cells and organisms they can infect. So to explore those differences a little more, we'll talk about the structural differences. So again, I said every virus at a minimum has these two things: the capsid protein coat and genetic material. So that's kind of the required minimum for a virus. In addition, some viruses have a viral envelope. So again, this is not something that all viruses have, but some have. That's going to make them structurally different. The capsids can come in lots of different shapes, and these are some examples from your textbook. The capsid can be shaped like a hollow tube. A virus called tobacco mosaic virus, which infects a lot of different plants, has a capsid that's shaped like that with the genetic material on the inside. They can have this icosahedral shape, which is what I think 16-sided. And we don't draw all 16 sides because that would be impossible, especially with my limited artistic skills. But it has many sides to it with the genetic material on the inside. And this is an example of there's the protein coat, the genetic material on the inside, and then a viral envelope on the outside. This is another sort of unusual shape. This is called a phage virus, and it has the icosahedral capsid, but then it has this unique sort of landing device, is kind of how I always think of it, that allows it to attach to bacterial cells because phage infect bacteria. So it's kind of funny to think about this for us, but bacteria get infected with viruses too. The other thing that I want to point out in the structure of viruses are these here. These are called spike glycoproteins. And they will stick out of either the capsid or the viral envelope, and they are important for attaching to the host. So fundamentally, they're going to help determine which cells this virus can infect, those spike glycoproteins. So again, just structurally, all viruses basically have a capsid and genetic material. Some of them have viral envelopes and spike glycoproteins, and those capsids can take on different shapes. Viruses also differ in their genetic material. So everything we've learned about so far in biology, the genetic material is always double-stranded DNA. And again, that's true for all things that are considered life. Viruses do not have just DNA as their genome. So some viruses do, they do use DNA, but some viruses use RNA as their genetic material. So their genetic material can be DNA, it can be RNA, it can be single-stranded. So again, this is a little different from what we've learned about before. It can also be double-stranded. It can be linear like our DNA, or it can be a circle like bacterial DNA. And it's always going to be the same for a specific type of virus, like phage viruses always have DNA as their genetic material. HIV, on the other hand, always has RNA as its genetic material, but it varies widely among the different types of viruses. And then the third way that viruses differ from each other is in their host range. The host range is the number of species or cell types that can be infected by that particular virus. Some viruses have a very narrow host range. So HIV is a good example of a narrow host range. It very specifically infects human immune cells. It doesn't transmit to other animals or other types of cells. The flu is a type of virus that has a very broad host range. So many animals get the flu, not just us. Pigs and chickens and horses and cats and dogs can all get influenza because it has a very broad host range. So this is looking at host range within cell types. So remember that host range is not just to a particular species, but even to one cell type within a species. And so if you look at some of the viruses that affect humans, you can see good examples of these. So ones that you've probably heard of, rabies, for example, very specifically affects nervous tissue, so the brain and the central nervous system. Influenza virus affects the respiratory tract. The rhinovirus, which causes the common cold, also specifically affects the respiratory tract. HIV, we talked about, is an immune system. It affects immune cells. Rubella, which is the measles. Epstein-Barr, which causes mono, all affect the immune system. Hepatitis B, rotavirus, Norwalk virus, all affect the digestive system. The herpes viruses, herpes simplex 1 is a skin virus, and that's why it causes cold sores. Let's see, herpes simplex 2, while it's related to herpes that causes cold sores, is the one that causes genital herpes, and so that very specifically affects the tissues in the reproductive areas. And then you have viruses that affect blood, and so of course Ebola is the one that we are all unfortunately very familiar with right now. So it's very important for you to be able to understand why a virus isn't considered living. And like I said earlier, you should really go back to the very beginning of the semester and go through that list of the properties of life and make for yourself a list of the ones that viruses do fit and the ones that they don't. And you might want to do that after we talk about the viral reproductive cycle. But that's a good way to both review from the previous parts of the semester and really make sure you understand viruses.